Gene expression is the realization of information potential encoded in the genome. A large portion of its process takes place before or during the stage of transcription. The many steps thereafter, mRNA processing, translation, and splicing, and protein localization add to the diversity and regulation of gene expression. The figure from the picture on the screen describes the life of eukaryotic transcripts following transcription and illustrates the mRNA as almost continuously processed, protected, translated, or degraded, mostly with specialized cells. As a consequence, mRNA is not usually present in the cell without RNA-binding proteins. Processing of post-transcriptional pre-mRNA takes place in the eukaryotic nucleus and bestows transcripts with features important for subsequent translation. Examples is a 5' cap structure, an intron-free open reading frame, and a precisely defined polyadenylated 3' end. Capping is the addition of GMP to the 5' triphosphate end of the transcript in the unusual 5' to 5' direction, and the subsequent methylation of the position N7 of the transferred GMP. This process is required for all cell viability due to its resulting protection of mRNA's 5' to 3' exonucleus activity. That initiation. When exiting the nuclear pore complex, the nuclear binding proteins are exchanged for the cytoplasmic translation initiation factor ELF4E. That initiation factor prevents the mRNA's denaturing by inhibiting the decapping enzyme DCP1P to the transcript's 5' end. The recruitment of ELF4E and other translation initiation factors to the cap enhance translation and promotes the engagement of ribosomal subunits with the mRNA. Central dogma, or Crick's central dogma, named after Francis Crick, who coined the term, can be broken down into a few major steps: transcription, translation, and replication, all of which helps the information in genes flow into proteins, from DNA to RNA to proteins. RNA processing factors appear to substitute the original transcription initiation factors on the transcribing RNA polymerase II, and by binding to the polymerase II elongation complex as it makes the mRNA, they couple transcription with mRNA processing. This loop is able to link translation and mRNA decay, and eventually translation initiation and termination on a circular mRNA. The next pre-mRNA modification takes place in intronic splicing, which uses spliceosomes, a macromolecule machine that contains 5 small nuclear RNAs and numerous proteins, to remove specific sequences, introns, from the pre-mRNA to transform the leftover exons into mature mRNA. After one final modification, which is the 3' cleavage, and an addition of a poly-A tail. The poly-A tail, by providing a binding site for PAB1P, enhances translation and stability of mRNA. Capping, splicing, 3' cleavage, and polyadenylation factors have all been associated with the C-terminal domain of transcriptionally elongating polymerase II. The next step in mRNA utilization is translation, which requires transport from the nucleus to the cytoplasm through the nuclear pore complexes. Prior to this, mRNA is bound to the heterogeneous nuclear ribonucleoproteins, which, while not considered a direct role in mRNA transports, shuttles between the two compartments, carrying the transcripted mRNA to the cytoplasm where the information it carries will be translated. Translation can be divided into 3 phases. During initiation, the translational apparatus is assembled onto messenger mRNA and recognized as the start site. Throughout elongation, the actual protein synthesis takes place, while in the last step, termination, the translation is ended and the peptide product is released, and the ribosomal subunits are recycled to start the process over again. Initiation is the first step toward translation of mRNA, and due to its complexity is also the site of extensive global and transcript-specific gene expression. There are actually 3 ways to initiate translation in eukaryotes: via the 3' end, by recruiting a ribosome to an internal ribosomal entry site, and by reinitiation of translation by post-termination ribosomes. The most common method is via the 3' end cap structure. This creates a bridge composed of the cap structure, the cap binding structure, ELF4F, and ELF3, and the 405 subunit. The initiation factor ELF4G also contains a binding site for PAB1P, which then allows the poly A tail to promote ribosome recruitment to the mRNA's 5' end independent from the cap structure. The 5' and 3' ends act to enhance translation rates, which is thought to be due to the ELF4G to the PAB1P interaction. Following initiation is elongation, in which the coding potential of ORF is realized and converted into a peptide sequence. While the ribosome moves 5' to 3' in relation to the mRNA, amino acids delivered by the amino-acylated tRNAs are joined by the formation of peptidal bonds, producing a polypeptide. This event is energy-consuming and requires GTP and amino acid tRNA to be sent to the A site of the ribosome of the highly abundant EEF1A. Recognition of the codon by the charged tRNA involves hydrolysis of GTP bound to the ternary complex. The EEF1A to GDP complex is then recycled by EF1B. After the transfer of the peptide chain onto the amino acid tRNA localized in the A site, another G protein, EF2, is responsible for the translocation of the charged tRNA into the P site and the transport of deacylated tRNA to the translocation of the charged to the E site. The next round then begins with the A site when the A site is loaded again with the ternary complex. The elongation rate is mainly determined by the amount of amino-acylated tRNAs available as well as the activity of various elongation factors. Because of its potential for processing, it is viewed as a single entity, and therefore a change in the rate of elongation would only affect the ribosome density of ORF, but that does not impose and change the rate of translation. Eukaryotes rely on an in-frame stop codon that is translated into a ribosomal A site, where this polypeptide chain factor is made of ERF1 and ERF3. ERF1 recognizes all three stop codons, UAA, UAG, and UGA, and mediates the hydrolysis of peptide from the tRNA in the P site. ERF3 recruits ERF1 and stimulates this reaction in a GTP-dependent, codon-dependent manner. As the amino acids are linked into the growing peptide chain, the proteins begin folding into the correct shape. The folding continues until the peptide chains are released from the ribosome as mature proteins. The correct folding process may require other proteins, called chaperone proteins. Occasionally, proteins can be further spliced. When this happens, the inside discarded section is known as intine. The final step of central dogma is replication, which transmits genetic information between protein and progeny. Replication is carried out by a complex group of proteins, which includes helicase, which serves as a zipper which serves as an unzipper by breaking the bonds between two DNA strands. Once the two strands are separated, a small piece of RNA called RNA primer is attached to the DNA by an enzyme called DNA primase. These proteins unwind the superhelix along with a double-stranded DNA helix, and by using DNA polymerase, its associated proteins copy or replicate the master template itself, so the cycle can start again in a new generation of cells or organisms. The central dogma is not really a dogma in the traditional sense of the word. Like all scientific theories, it is modified as we learn more about the details of its processes. The biggest revolution in central dogma was the discovery of retroviruses, which transcribe RNA into DNA through the use of a special enzyme called reverse transcriptase, has resulted in an exception to the central dogma, where it goes RNA to DNA to RNA to proteins. Also, some virus species are so primitive that they only use RNA to protein, having not yet developed their DNA. With the discovery of prions, a new exception to the central dogma has been discovered, protein to protein. That is, proteins directly replicate themselves by making conformational changes in other proteins. Although retroviruses, certain primitive viruses and prions may violate the central dogma, they are not considered technically alive. and thus the rule all cellular life follows essential dogma still holds true.